Well, hi everyone. Welcome to MUMS seminar, MUMS fortnightly seminars. This is the one we're running in week 6, and today I have for you two brilliant applied mathematical biology students of Jennifer Flegg, and together they have volunteered to give talks on some of the work they've done, and yeah, I hope they can take it away. First up, we have Yong See Foo, who's a master's student. Would you like to introduce your talk?
Yeah, so I'll introduce myself a bit. So I'm a master's student. This is my last semester. My specialization is in statistics and stochastic processes. So over sort of this semester and the last 2 semesters, I've been working with, as you heard, Jennifer Flegg on this project about mapping malaria mutations with Gaussian processes. So today I'm actually going to try to keep the maths more on the simple side, I would say, and sort of introduce some ideas about Bayesian statistics instead. So let's get started with a bit of a motivation to why we care about mapping malaria mutations. So malaria is a really widespread disease that still is quite a pain in a lot of developing countries. It still kills almost 500,000 of people a year. And so there's a lot of work done to control the spread of malaria. Now, if you want to stop malaria from spreading, you sort of have 2 things that you want to do. One is you want to reduce the number of infections that you will have from malaria. And 2 is that when people do get infected with malaria, we want to treat them as well as possible, so that 1, they wouldn't die, and 2, so that they can't further transmit the disease if you treat it adequately. Okay, now these 2 things are both complicated by one particular factor that I'm going to be focusing on. Of course, there's a lot of other factors, but one factor I'm looking at is drug resistance. So what that means is that when someone gets malaria, you would want to try to cure it with a drug, or even if they don't have it, you might sort of have a preventative drug that some people would take as well. Now, when you use this drug for a prolonged time or in a certain region, as you might know of COVID-19, in this case, we have malaria parasites, you will get variants. They will mutate, and in this case, they might develop mutations that make them more immune against certain drugs, and we call that drug resistance. Then once the drug doesn't work anymore, then you can't treat those people infected with that drug-resistant malaria, and then your preventative treatments also don't work as well. And so my work is pretty much about in which areas do we have issues about drug resistance. So let's look at why we talk about the spread of drug resistance. Let's say that you have some mosquitoes, because malaria is transmitted by mosquitoes, and say these 6 mosquitoes here are all infected. And 1 in 3 of them, shown in red here, have the drug-resistant malaria, right? And the green ones also have malaria, but they're not drug-resistant. We have 1 in 3 mosquitoes here that are infected with malaria with the drug-resistant version, right? That's going to cause more problems. And then the other 4 sort of have a more regular malaria infection. When they bite people, these people can become infected with malaria. And so let's say 1 in 3 people who get infected also have this drug-resistant version of malaria. Now, these people would then seek treatment, so they would take a certain drug. And then you would expect, in terms of proportion, the ones who have the normal version, not the drug-resistant version, most of them should recover. So let's say in the 4 of them here, 3 of them recover. But then because the drug-resistant malaria is stronger against the drug, so maybe only 1 and 2 of them recover. So you have sort of this situation. Okay, now how does malaria continue to transmit? What happens is that when you have other healthy mosquitoes who bite these infected people, then the mosquitoes become infected with malaria, and it goes on. But now that we just focus on these 2 people who are left infected, and of course, in reality, that's probably still hundreds of people, but about half of them this time have the drug-resistant version. And so your mosquitoes, you have 1 in 2 mosquitoes now that have the drug-resistant version in red. So when you compare with before, what has happened is that the prevalence, or like the proportion, I'm going to use the word prevalence a lot, the prevalence of drug-resistant malaria increases. That's the basic idea of what we talk about the spread. And so if you start from a certain location, this will sort of spread out and the levels of drug resistance will increase. So there's some motivating questions we have. One is, how high is this prevalence? Can the current drug still be used? If it's too high, then it's going to fail too frequently, and you don't want to worsen the situation. Another question could be, how is it spreading? Like, where is it spreading to, and how fast is it spreading? So we think about what's the geographic spread of this resistance over time. And of course, to answer all these questions, you need to collect data. And collecting data can be really expensive, could take a lot of time, and so on. So your resources are limited. So the last question could be, say, where do we need to collect more information or more data? Okay, so now we're going to talk about the data a little bit that might help us answer these questions. How it works is that people will collect blood samples from different sites, different villages, across different years as well. So what we have here is what we call spatial temporal data, data from different locations at different times. And what kind of data it is? Well, it's count data. Because what happens is that you take, say, 100 blood samples, and then you test them genetically and see what kind of mutation they have, and you sort of know which types of drug-resistant mutations you're looking for. And for now, I'm going to assume that like each malaria blood sample is either going to have no mutation, or maybe it has mutation A, or has mutation B, and so on. It wouldn't have two mutations at the same time for now. So I have data from 2010, 2011, 2012, all the way to 2019. I'm just going to show you sort of 4 years. So you can see that every year they take data from different places. This is Southeast Asia, but they're different villages. The sizes are different because the sample size is different. And sometimes your data is a bit denser, sometimes the data is sparse. Most of the time it's sparse. Hopefully you can tell. And so I've just sort of drawn the percentage of parasites we observe to have some mutation. Now this data is pretty sparse. There's a lot of areas where we don't know what's going on in terms of drug resistant levels. So what we want to do is regression. So we'll talk about regression a bit later, but the basic idea is that we want to color in these maps. We want to sort of know what's the level of drug resistance in the other areas where we don't have the data. Okay, so that's sort of the problem set up so far. That's the introduction. And that brings me to my outline. So how do we answer those 3 motivating questions we had before? And what we're going to talk about is estimates and uncertainties, sort of a general idea in statistics. The next natural question would be like, how do you obtain estimates and uncertainties? And the way we're going to do it is to use Bayesian inference, and in particular on the spatial temporal data. And lastly, how do you handle this spatial temporal data? Again, there's a lot of methods, but the method I'm looking at is something called Gaussian processes, and we'll see how they fit into a Bayesian hierarchical model. So those are sort of the 3 things. The first 2 probably will go by quickly, and the last bit will take me a bit longer to explain. So let's go on. So we're going to talk about why we care about estimation and uncertainty. So here's a toy problem that people who teach probability really like to talk about. So say someone gives you a coin, and you want to figure out whether the coin is fair. That means, does it turn up with heads 50% of the time? Okay, so you probably flip it a few times and try to make some sort of estimation of Q. So let's say you flip the coin 10 times, and you get 7 heads. And I ask you to estimate Q, you probably would say 0.7. Okay, and then maybe someone else, they flip the coin 100 times and they get 70 heads. And then you say, okay, I'll guess Q is still 0.7, right? But you sort of know that from the first example, you might go, well, the coin might still be fair. I just got 7 because of luck. But in the second case, you know that that 0.7 is a bit more certain. Like you're more certain that the coin is not fair anymore because you've tossed it 100 times. So something really basic, but this sort of shows you that an estimate is not enough. Because in both cases, you say 0.7 as an estimate. You care a lot about uncertainty as well to make good conclusions. Okay, so let's say instead, well, maybe not that yet. So how do you address uncertainty? A lot of people will be familiar with the idea of confidence intervals, but confidence intervals are actually pretty messy to interpret. So we're not going to go down that path. Instead, I'm sort of going to define uncertainty as the spread of likely values of Q, that's a probability of heads, after you observe some data. Okay. So perhaps after you do 7, you get 7 heads out of 10 flips, you might say, okay, like Q is most likely around 0.7, so that's a peak, but then it's quite spread out. Like I'm not really sure what the exact value is just yet, because I only did 10 tosses, 10 flips, okay? Whereas if you have 100 flips, then you can still have a peak around 0.7, but it's much narrower. You have a much smaller uncertainty because you're a lot more sure about that estimate. So you can see high uncertainty on the left, and low uncertainty on the right. So how do we sort of like get these sort of spreads, these graphs? That's where the Bayesian statistics will come in. Okay, so I'll talk about Bayesian inference now. And again, we'll start with another sort of toy example that perhaps people are really familiar with. So this is about rapid antigen tests for COVID-19. I think at some point, many of us will have done a test because we got symptoms, and then we got a negative result, but we're still a bit worried, like, am I actually still infected? But I sort of just got a negative result because the test is not perfect, things like that. That's naturally something you will calculate with Bayes' theorem. And I think a lot of people have seen Bayes' theorem before. So this is sort of the situation I was just talking about. You're really interested in knowing, I know that I have a negative result, but what's the chance that I'm still infected? And then you sort of have all these things to calculate through. Especially, you can sort of imagine what each of these mean. Probably infected is just, given a random person, what's the chance they're infected with COVID-19? So maybe like 2% or something like that. Probably lower than that, I think. Whereas here, this is sort of the second term. It's about how good this test is, right? If you're infected, how low is the chance that you get that wrong negative result? Okay, and we're going to try to generalize all of these things a bit. The bottom thing is the chance that you get a negative result overall, whether you're infected or not infected. So you sort of have to take an average of the two, whether you're infected or not. And so that bottom bit should be some sort of sum. And if the law of total probability sounds familiar to you, that's what it is. So we'll go on for now. How do you sort of generalize this? Well, let's say like you want to talk about both not infected and infected, and both negative result and positive result. Then you can sort of write Bayes' theorem in this way instead. But what I'm going to point out is let's think about model and data in a general sense and try to fit this into this scenario here. What your data is the result you got. That's what you can observe. I got a positive result, I got a negative result. That's data. You also have what you can't observe directly, which is, are you infected? That's sort of an unknown parameter for now. You might be infected, you might not. But we wanted to sort of say, what do you think the parameter is given the data you observe? And the last thing is that thing in the bottom there is always going to be some sort of messy sum that you have. So I just sort of rename all of that. We're going to make this generalized now. You have this situation going on. And we actually have names for each of these. So that thing there is what we call likelihood. So you might remember this was what's the chance that I get a negative result even though I'm infected? So how likely is that? So that's why we call it likelihood. It's some evidence you have about the parameter from observing the data. We also have what we call the prior. And in general, that means the initial beliefs about the parameter. So that's sort of like out of everyone in Victoria, like how many people have COVID-19 should be pretty small. So I believe maybe I have a small chance of getting COVID-19. But maybe you've just been at a big event, then you might want to up this value a bit. So I'll talk about this thing about prior choice a bit later as well. What you're really interested in is in this thing called the posterior. And that's your updated beliefs about the parameter after observing the data. And there's always this messy scaling factor. There's a technical name for it. I won't refer to it much. Okay, so people like to, again, use more notation. Y is your data. Data is your parameter. And this is sort of the same thing again, but shown in this formula instead. All right. So how do you do Bayesian inference? Again, as a summary, if you look at the top bit, you want to take your initial beliefs about the parameter, that's the prior. You combine it with the evidence from the data, that's a likelihood. You will have to do some sort of scaling because of the bottom bit, and it doesn't have the theta, that's why it's just a scaling factor. And then you will get to update your beliefs about the parameter, that's the posterior. Okay, so the scaling factor, I said before, it's a messy sum, but if your parameter is continuous, then you have to do some sort of integral instead. So it's some messy integral. We're not really going to talk about that too much. And so I'll move on for now. All right, so if we go back to the point example, the parameter we have is Q, the probability of hits. We don't know what that is. But we do know the number of hits we observe. So say 7 hits, that's the Y value here. Okay, and so this likelihood thing would be a binomial distribution if you know what that is. And so you should choose a prior for what your beliefs are about this Q parameter, and then you combine it with data and yada yada and so on. So let's talk about choosing a prior. One way to choose P of Q, the prior, is to say, well, I think all values are pretty likely. Like this coin could be 0 chance of hits to 1 chance of hits, anything in between, uniform. Okay. You can also be a bit more subjective and say, well, it's really unlikely that a coin is going to always be heads or always be tails. So let me actually sort of avoid the extremes and concentrate a bit more in the 0.5 area. So you can sort of make your prior flexible according to what you believe. All right, so let's say we take that second prior, and then we take this likelihood bid, which is you see 7 hits out of 10 points, 10 flips. You combine these, and that's how you can get this curve. You will multiply these two functions. So this function of this curve, this function here of Q, you will have to divide by that messy integral, and I've done that here, and you can get this graph. Okay, so we can summarize this in the Bayesian model. The Q parameter is going to sort of generate your Y. Q is unobserved and Y is observed. So how does this work for malaria mutations? Well, there's some differences. We don't just have one Q here, but we have multiple sites. Right? So maybe that's, and more than that, we don't just have heads or tails. We might have no mutation, mutation A, mutation B. That's multiple categories. The likelihood will be multinomial instead of binomial. And so I can draw that diagram again, but maybe in this way. The box here just means that you have to repeat this n times, and the bold face here just means they're vectors now, instead of just a single number. The scaling factor, the integral, is going to be hard to compute. And so there is a technical detail here that we don't calculate the integral. We actually simulate from the posterior, but that's sort of an advanced topic that I won't really talk about. If you're interested, that's the reference you should look up. Okay, but the most significant thing for malaria mutations is that we have spatial temporal dependency. That means you have P's, probabilities from different locations, but if the locations are close together, then the P should be similar. Whereas if the locations are far apart, then your P might not really have that dependency. And so we need to find a prior to express this initial belief about the parameters P. And that's where we're going to talk about Gaussian processes now. So here we come back to regression. A pretty simple example again to start with. You probably know about linear regression. Your data looks like it surrounds a line. You fit a line to it. Pretty all right. But what if it doesn't fit a line? Then you might have to sort of guess and go, oh, maybe it's a quadratic. So let me try to fit something like ax squared plus bx plus c. And you fit a quadratic. And you go, some points are a bit off. So maybe this is not an accurate fit enough. And then you go, okay, so make it a higher order polynomial or something like that, so that I can get these things accurately. Well, you can, but then if you fit a higher order polynomial, you might get something that just doesn't look realistic enough with all those weird bends and so on. So what you want is something sort of in between, right? You sacrifice some accuracy, but you make it look realistic. And that's what you can do with a Gaussian process. So I'll get into the details about the Gaussian process now. Oh, sorry, one more slide before that. So why does this help us? Well, that's because these dots you see here could be those dots I showed you before, right? And to be able to fill in that curve is pretty much to color in those maps. So that's sort of the analogy why we care about this regression problem. So now I'll go back to talking about regression. So here's the general way you would spell it out. Each input is associated with some sort of output, f of x, but then there's some noise that sort of corrupts it. That's why we see things being off the curve. Okay, so you can write it down like this. The epsilon there is just a noise. And so in your linear regression and things like that, we call that parametric regression. You assume something about f, maybe it's linear, maybe it's polynomial. But Gaussian processes are different. They're what we call non-parametric regression. You actually don't assume a predetermined form for f. So the key idea instead is that Gaussian processes is a distribution that gives you functions. It's a bit different from, let's say I have a random number generator between 1 and 100. You just keep getting integers, 51, 39, and so on. But then for Gaussian processes, every time you get something, you actually get a whole function. Okay? This function has a particular property. You can't just get any whatever function you want. F of x and f of x dash, they're going to be correlated. And the closest x and x dash are, then the more correlated they are, because they have to be quite similar in value. Whereas if x and x dash are further apart, then you have less correlation, a bit more flexibility of what those values can be. They're less dependent on each other. So you might say that the value here and the value here are quite correlated because they have to be close. But the value here and the value all the way there, there's not as much correlation between the two because they're far apart. Technically, you can go define some mean and some covariance. That's if you know what these terms mean. And basically, the mean function is what determines the average height of your function, I suppose. And the kernel is what determines the shape of the function that you will get. There's some notation here that you might see later on. And we're also going to assume that the mean is 0. It turns out that different kernels will give different properties for x. I'll just show some examples. This is the most standard one you will have. You get pretty smooth functions like these. If you multiply that by 2, then your functions are going to vary in a larger magnitude. If you change the number inside the fraction, you can make these functions vary not as frequently. They're a bit more flat. Or if you make that constant different, you can get them to vary really quickly. If you don't use this exponential thing, you use something else, you can get other ways for them to fluctuate. This one seems to fluctuate like in a small range, but on a small scale as well. And you can even make them not smooth if that's what you think it should be. So these are all the different ways you can choose a prior, a different initial belief. So we're running short of time, so I'm going to sort of speed up the last bit a bit. So what we've talked about so far here in this part can be summarized by these 3 equations, and also by this diagram. So you sort of just see that the shaded things are the things you observe, and then it will be like this sigma generates this noise, but then this Gaussian process and this noise will generate what like data you observe. So you sort of can draw this model, I suppose, and this will come back at the very end. So again, we still have this idea of prior and likelihood, which is something about the data, evidence from the data, and some initial belief about your parameters. In this case, the parameter is f. So putting things all together, we're getting to the end. We have H categories in our malaria example. And then the likelihood would be the multinomial likelihood I mentioned, some probabilities that generate these counts for you. So that's this picture you've seen before. And now prior is a bit complicated, right? You have these probabilities that should have some dependency if they're close together, and then you want to use Gaussian processes. Complications are x is no longer just one dimension, it's got like longitude, latitude, and year. You can't just have one Gaussian process because you have h different probabilities, so you need like h different functions instead. Gaussian processes don't have a bound, but your probabilities have to add to 1. And so then there's some complicated way that I want to explain to convert all of these f's, which don't have any bounds, into p, so that all your pij's would sum to 1 across the j. Okay. And so this gives you us a more complex prior. That was what we had before in the Gaussian process example. And now all the changes I mentioned are here. This is still the mean and the kernel. A lot of things are bold because they're now vectors. And this used to be y, something you observe, but now it's just a probability p that you don't observe. And so we take the prior, we take the likelihood, and then you sort of just fit these two Lego pieces together, and you have your full model. And this is actually a diagram pretty much straight out from my thesis. Okay, now that you have a model, you can go find the posterior distribution. That's what we wanted to know, which is after you observe all these y's, what do you think p is? What's the estimation and what's the uncertainty? And you can even do that in places where you haven't observed the data yet. Okay, so that's why you can see it's not just the dots that we color in, but everything else as well. I can't draw you a density curve for each of these pixels like before. So I have to show you some summary instead. And what you're going to see, the color is sort of the mean, and how transparent the color is the standard deviation. Like the more transparent it is, the more uncertain we are about it. Then I can show you part of the graphs. There's actually more graphs down, but here's just some of them. And it helps us answer those questions we started with. Can the current drugs still be used? Maybe that's something to be answered by looking at the estimates here. Maybe you pick a threshold and say that the drugs should still be used here, but not there, depending on how prevalent the mutation is. You can talk about how does it spread. You can, for example, this one seems to be spreading southwards when you compare there. And you can talk about how fast it's spreading and so on by looking at these maps changing over time. We can also talk about where you need to collect more information just by looking at which bits are the most transparent. That's sort of where you have the most uncertainty. So wrapping up, I hope you learned a thing or two about Bayesian inference or about Gaussian processes. There's a lot of other extensions to this actually, like this is part 1 of 3 of my research. I really don't have time to talk about other things, which is how you can make these Gaussian processes, the different Gaussian processes, dependent as well. And also what to do if people don't fully report all their data, which happened a lot when I looked at the data. So special thanks to my supervisor, Jennifer Fleck, who has been helping me a lot with getting this project together and sort of developing the ideas behind it. And if you're really interested in this stuff, I will give a talk in week 12 later on. Or if you want to just ask questions to me or keep in touch about this stuff, you can contact me here as well. So that's where I'll end.
Thank you very much.